This is video number 2 in the series from chapter 4 of your textbook, Protein Structure and Function. Video number 2 covers the topics of the hierarchy of protein folding and some common folding patterns that we see in proteins and how the different kinds of folding can affect the functionality. Here's an illustration that shows schematics of a number of very common proteins. We can see the diversity of shapes and sizes, and if you know the functions of any of these, such as collagen or hemoglobin or any of the others, you know that their functions are very different, which reflects the fact that they're shapes. There are different ways that diagrams will represent the shape of a protein and its folding patterns depending upon what parts of the protein and what aspects of the folding patterns you're most interested in. So you can see the examples to the right of this slide. The primary structure of a protein would be its amino acid sequence. But if we go up a level in the hierarchy to the secondary structure, what we're talking about would be interactions between adjacent amino acids or among several amino acids that are in close proximity to one another. There are two very common secondary structures which are pictured to the right of this slide: the alpha helix at the top and the beta sheet at the bottom. Here we can look at the alpha helix a little more closely. What I want you to particularly note, in addition to the helical pattern, would be these hydrogen bonds represented by the parallel pink lines. Hydrogen bonding, as we discussed on the previous video, can happen between backbone atoms of the amino acid chain, specifically between nitrogens and oxygens. The helical structure brings nitrogens and oxygens in close proximity to one another in such a way that they stabilize the helix. Helices can be right-handed or left-handed. They form readily in proteins, and they also form in other biomolecule types. You're probably very familiar with the double helix that would be the structure of a DNA molecule. Here's an example of an alpha helix of a membrane-bound protein. In this case, because the helix exposes its side chains on its surface, hydrophobic amino acid side chains can interact with the hydrophobic tails of the phospholipids that make up the membrane's bilayer. This allows the protein to be embedded in such a way that it's very stable within the environment of the membrane. Two alpha helices sometimes wrap tightly together to form a very strong and very compact structure called a coiled coil. In this case, hydrophobic R groups along a band on the structure, represented in green in the middle illustration, interact with one another between the two alpha helices. Because they exclude water molecules from this space where the two helices interact, they can create an extremely compact and strong structure. If we turn our attention now to the beta sheets, what we can see is that a beta sheet consists of strands of amino acids that loop back adjacent to one another, and the hydrogen bonds that form between backbone nitrogens and oxygens stabilize the interactions between adjacent strands. On the left of this illustration, we can see that adjacent strands may be oriented in opposite directions to one another, as in part A, or they may be parallel to one another, as in part B. We call both of these beta pleated sheets, but A would be anti-parallel, B would be parallel. To the right side of the screen, we can see an example of a protein molecule that consists of beta pleated sheets. Silk is both very strong and very flexible, and these properties are due to the types of interactions that occur within and between beta pleated sheets. So we've talked about primary structure and we've talked about secondary structure. Sometimes several secondary structures may join together to form a region of the protein that's independent, partially independent from the rest of the protein. We would call this a single protein domain. In this illustration, the middle of the slide shows us a group of several alpha helices and a beta sheet that are clustered together. We could expect this single protein domain to have a function that contributes to, but is not the entire function of the protein as a whole. On the right, we can see an entire protein molecule, one single polypeptide chain folded with several levels of hierarchical structure. We would call this the tertiary structure of the protein. Here are some illustrations of secondary structures grouped together in different ways. Numerous alpha helices and beta strands in conjunction with one another, and several beta strands interacting with one another. Now, in addition to alpha helices and beta sheets and protein domains, sometimes proteins have regions that remain unstructured. These unstructured regions are useful for the specific purpose of the protein that has them. Like, for example, on the left, in red, we can see a protein whose job is to bind other proteins. The unstructured protein, when it is not associated with its binding partner, is very evident. But we can see that it's able to bind in very specific ways with its binding partner and thus alter the function of the binding partner. In the middle of the screen, we can see a protein that has a tethering domain. You can see the unstructured region in red, and you can see the blue tethering region at the end. The flexibility of the unstructured region allows it to bend and move from one place to another, so it can bind, in this case, within the protein itself to a particular binding region under certain circumstances. If we go to the right illustration, what we can see is that tethering domains are also extremely important in connecting proteins together. So the tethering domains allow the protein to interact with other proteins and can serve as a scaffolding protein, such as you may remember discussing when we talked about DNA organization in nuclei. And we'll be addressing that issue again in the coming weeks. Proteins have many different functions as a whole, but a lot of the thousands of proteins can be grouped together according to similarity of function, which of course corresponds to similarity of structure. So these groups of proteins we would call families. They have similar shapes and related functions. They may be found in different cell types or in different kinds of organisms. One protein family that we'll use to illustrate the concept of protein families is the serine protease protein family. If you look in the center of these two proteins, elastase and chymotrypsin, what we can see is that their active sites are virtually identical, as illustrated with the red line drawing in the center. However, if you look at the folding patterns of the polypeptide chain, what you can see is that the loops and the ways where it folds back on one another, though they are similar in most places, they're far from identical. This would be typical when you would compare members of a protein family.